What's going on, ladies and gentlemen? What's up? Welcome to the StormCast episode 6. We're live on YouTube, Rumble, Twitter, Facebook. So follow up on those to be notified when we go live. Shout out to the Spotify, Apple Podcast, Facebook, Amazon, iHeartRadio podcast listeners. We're everywhere you can listen to podcasts here at the Green Room in the Stormy studio. Well, today we got blue lights again, but it's my little studio room. I'm Stormy, your host for the evening, and in this episode, we're looking at the latest news studies in the world of neuroscience and reacting to some videos and all other topics. So let's just jump right into it. Today is February 10th, and it's a warm and cloudy and 91 degrees outside. Hope everyone is having a great week. As we enter this beautiful weekend, I just want to say I've been getting into a lot of fantasy football lately. As we all know, preseason has already started, and NFL season is right around the corner. So this year I'm going to be doing some betting. So we'll see how it turns out. I've been, doing a lot of mock drafts, practicing on my picks. I got my strategy. I got one of the best strategies in fantasy football. I'm not gonna lie. I think I got it down to a science at this point, and this year we're putting some money on the line. So if you guys aren't tuned into my Rumble, I do a lot of my sports betting streams on there. So go follow on Rumble at rumble.com/stormyfriends. I'll be doing a lot of fantasy football and other types of streams on there. As of lately, I've been having a lot of house renovations going on, trying to make this house accessible. As you know, I just bought a new house, new crib. So when you get a new house, you gotta get the house, how you like it. It's not always gonna be perfect. I mean, a lot of it was already set pretty good, but I had a couple renovations I needed to get done to make it like completely accessible. So we're almost there. We're right around the corner. A lot of things have been getting done. The bathroom is looking nice. We took the tub out. It's all rolling at this point, and we just gotta add some other additions. So this coming up week, I should be taking showers in my new shower. So I'm excited about that. So I did get some stem cells recently. It's been about 2 weeks. I'm gonna give you guys a little update. I'm feeling good, feeling strong, still, still working out, getting back into it. My workouts have been real smooth and my inflammation has been real low. And so I've just been trying to continue to work out, do as much as I can physically, and do a lot of meditating and focusing on it, the healing process. And so I'm excited about workouts now, and they've been helping out with workouts a lot. So I think I'm gonna continue to do stem cells in the future, and I do have another injection coming up here soon. So I'll be dropping another video on that. This process has been wild, and I'm just excited about the future of healing and therapies that are continuously evolving, and who knows what the future looks like for people with SEI or disability. So I just want everyone, if you're joining in, to drop a comment and a W in the chat. If you're watching live on either Rumble, Facebook, or Twitter, or actually we are on YouTube, Rumble, Twitter, and Facebook. So go follow on there, follow up. So we're gonna get right into these little topics that I wanted to talk about today. I saw some pretty interesting news coming out. So I'm about to share my screen here with you guys, so we can all be on the same page here. Let's see how. Okay, so I'm gonna have to share my screen down here. That makes sense. Yeah, guys, we have been continuously grinding and working on new content ideas and topics to bring to Stormy and Friends and all related channels. So we're doing a lot behind the scenes, guys, and thanks everyone for joining in on today's episode. We're about to jump right into the first topic of the day. I got it shared all the way down here. I'm doing all of this by myself, guys. It's a one-man studio today, so most of our episodes are obviously, but being able to talk and do your own behind the scene production can be a task sometimes, but either way it goes, guys. Today's first topic we're going to talk about is illuminating brain inhibition, and this is a article from January 24th talking about how researchers developed a groundbreaking technology to track when brain cells deactivate, a key process known as inhibition. This technique allows a deeper understanding of normal brain functions and disorders like depression, PTSD, and Alzheimer's. By using optogenetics and identifying changes in the pyruvate dehydrogenase or PDH protein, the team can now observe how the brain conserves energy by rapidly shutting off the brain protein post-neuron firing. This discovery opens new avenues for studying brain cell inhibition and its role in various diseases. So this is going to be interesting, talking about how this new technology tracks the deactivation of neurons, shedding light on how the brain regulates activity through inhibition. The study revealed rapid changes in PDH protein, key to energy production, immediately after brain cells stop firing. This technique can be instrumental in understanding and potentially treating a range of brain disorders and metabolic diseases. Nice. For decades, scientists have studied the intricate activity patterns in human and animal brains by observing when different groups of brain cells turn on. Equally important to understanding the brain and related diseases, however, is knowing how long these neurons stay active and when they turn off again. Now, scientists at Scripps Research have developed a new technology that lets them crack when, after a burst of activity, brain cells shut off, a process known as inhibition. This technique, published in Neuron on January 23rd, 2024, provides a new way to study not only the normal functioning of the brain, but how the brain's off switches may go awry in normal behaviors, as well as diseases and disorders including depression, post-traumatic stress disorder, and Alzheimer's. It's generally agreed that the inhibition of neurons is really the major way the brain is regulating activity, says senior author Li Yi, PhD professor and Abide Vividan chair at Scripps Research. Scientists have been looking for a way to look at inhibition on a more trackable way, and until now, few have found it. For pioneering the new approach, Ye teamed up with John Yates, a professor of molecular medicine at Scripps Research. They wanted to study how brain cells changed when they were actively firing, emitting an electrical charge to pass messages to their neighbors, compared to when they are done firing. The scientists used optogenetics, in which cells' activity can be controlled using the light to repeatedly activate and inhibit the cells. Then they measured levels and characteristics of different proteins and their modifications. They identified that one protein, pyruvate dehydrogenase, or PDH, was very rapidly changed immediately after brain cells were inhibited. Neurons are firing. You need a lot of energy, and this PDH protein is involved in producing that energy, explains Ye. But the brain really wants to conserve energy, so when a cell is done firing, we found that the brain rapidly shuts off the PDH protein. This happened much faster than anything else we saw in gene expression. To shut off PDH, the researchers found cells add molecular tags called phosphates to the protein. Ye and his colleagues found antibodies that only recognize this inactive phosphorylated form of PDH. To test whether levels of phospho, phospho, phosphorylated PDH or PPDH could be used as a proxy for brain cell inhibition. It's a tongue tie right there. Phosphorylated. Say that 10 times fast. Ye's team used these antibodies to measure PPH in mice that had been given anesthesia. Nearly the entire brain lit up with high levels of PPH, correctly showing how most of the brain is inactive during anesthesia. The group also studied levels of PPH when animals were exposed to bright light that was then shut off. Brain cells in the visual cortex responsible for vision had low levels of PPDH when being exposed to light because the active form of PDH would be required to give these cells signaling energy. But high levels of phosphorylated protein immediately increased after the light was shut off. Ye's group also used the new technique to study a less understood process, how the brain turns off the feeling of hunger after a meal. They showed how brain cells related to appetite shut off when a mouse starts to eat. Those findings could have implications for better understanding appetite, obesity, and some weight loss drugs. More broadly, the PPDH antibodies could be used to compare levels of brain cell inhibition in people and those with a variety of brain and metabolic diseases. There are a lot of questions that this technology can help us answer, says Ye. If the brain can't turn cells off, or if they've turned off faster or slower than usual, what happens? How does the inhibition of neurons play a role in different diseases? Ye and his colleagues are continuing to fine-tune the use of PPDH, but they say that other researchers are already using this technology. The antibodies used to measure PPDH are commercially available. In addition to Ye and Yates, authors of the study, phosphorylation of pyruvate dehydrogenase inversely associates with normal neuronal activity, include Dongyang, Yi Wang, Tiabano Qi, Zi Zhang, Liu Shin, Jinghei Ma, Zhugian Peng. So a lot of sounds like Asian or Chinese, perhaps. Researchers went into this at the study of Viteen Augustine of Scripps Research and Oscar Christian Gonzalez and Louis de Lassay of Stanford. So it's a pretty big project. I think it's interesting finding out how brain cells like are turned on and off and how they're controlled with the brain. Let's share the next screen. Where we at? Trying to pull this next screen up for you guys. Since we got 6 people live joined in across YouTube, Twitter, and Rumble. Shout out to everyone in the chat. We got a video about how brain inhibition works, but so we're gonna see it, some optogenetics. And this is optogenetics, controlling the brain with light, posted by Midtech TV. This is 13 years ago, ladies and gentlemen. Controlling the brain with light.
The human brain may not look like much, a couple of pounds of pink, wrinkled tissue, but the last 100 years of neuroscience have shown that the brain is an incredibly densely wired computer made out of hundreds of billions of individual cells called neurons. Neurons communicate with each other using both chemical and electrical signals. When a neuron is activated by chemical messengers, it produces an electrical pulse that results in similar chemical signals being sent to other neurons to which it is connected. Neurons come in thousands, perhaps tens of thousands of different kinds, each with a unique shape, a distinct molecular composition, and a unique pattern of connections within the brain. For example, these cells, the basket cells, have small input branches and output branches, and they release pulses of chemical that inhibit their targets. Whereas these cells, the pyramidal cells, are large and project long distances, and they excite the cells that they target. If we could figure out the role that each of these cell types plays in the brain, we could understand how they work together to create thoughts, emotions, and behaviors, and how flaws in specific cell types may lead to devastating brain disorders like schizophrenia, Parkinson's, and depression. Now, MIT researchers are working to develop tools that allow us to do exactly that. It is now possible to precisely control the activity of specific neurons, and therefore understand how each neuron contributes to brain function. Remarkably, these high-tech tools have come from microorganisms, such as this pond algae called Chlamydomonas. Chlamydomonas needs light for photosynthesis, and it swims toward light with the help of a small organelle called an eye spot. The eye spot
And just look at that organism. If you're watching the video podcast on YouTube, you see that this thing looks like a complete alien. And this is one of the organisms that are here on this earth. Like, it's insane. It's like a bubble with two long, stringy legs. Oh, man.
contains a light-sensitive protein called
Insane.
channelrhodopsin, which responds to blue light by moving positively charged ions across the eye spot membrane, causing the voltage of the eye spot to change, a bit like a battery being charged by a solar cell. But neurons are also electrical devices, and their voltage would also change if channelrhodopsin could be inserted into their membranes and then illuminated. So, researchers took a piece of DNA that encodes for the channelrhodopsin protein. Then, they put it into neurons, using a safe and effective method for delivering a gene, the same kind being used in human gene therapy trials. The neurons expressed the DNA, manufacturing copies of the channelrhodopsin protein and installing them in their membranes. As predicted, the researchers were now able to activate these neurons using light, without affecting any of the neighboring neurons. This technique, called optogenetics, allows researchers to drive the activity of specific neuron types to see how they affect brain activity and behavior. For example, researchers can deliver the piece of DNA that encodes.
So they're using light to trigger different parts of the brain in order to elicit a certain response that the brain is responsible for.
For channelrhodopsin into a set of basket cells, shown here in green. Then pulses of blue light activate only the basket cells, while the other cells, for example the neighboring pyramidal cells, are not affected. So, if a specific brain disorder is associated with dysfunctional basket cells, it would be possible to augment the activity of these cells and possibly repair brain function. And these tools don't just activate neurons, they can silence them as well. Archirhodopsin and halorhodopsin, for example, are proteins that are sensitive to orange light and produce the opposite effect to channelrhodopsin. When researchers deliver DNA that encodes for one of these molecules into a specific set of cells and shine orange light within the brain, they can turn off just the selected set of cells. By combining these
Why orange light, though?
tools, along with others now being developed, researchers will be able to control brain activity.
It's currently pouring down rain outside now. Insane. I don't know if you guys can hear that, but it is coming down outside. Crazy weather we're having.
With the degree of precision that would have been unimaginable a few years ago. Optogenetics is currently being used in animal models to understand how normal circuits work, but it may also lead to completely new types of prosthetics for treating a wide range of human brain disorders.
Wow. Okay. So that's a little bit about how optogenetics works. So obviously it's a topic that's been studied for quite a while now, but it's still fascinating to understand a little bit on how we can control the brain differently using different techniques and behaviors. Right now we're going to pull up another video that I want to share with you guys. It had an interesting title. It's explained optogenetics. Hold on, bear with me. We are going to go right now. So let's get right into this video. Optogenetics explained by Massachusetts Institute of Technology, or MIT.
The brain is made of maybe thousands of different kinds of cells called neurons that are built into very dense intermeshed networks that communicate. Each of these neurons computes using electricity. This cycle implements behavior and thought and emotion, all these different kinds of things. We also think that deficits in these electrical computations underlie many brain disorders that affect over 1 billion people around the world. In optogenetics, what we're doing is we're putting molecules that convert light into electricity into neurons, the cells of the brain. Then when we shine light on those neurons, light gets converted to electricity and allows us to turn on or off those cells. The goal here is to find a way to control the electrical activity in some cells and not others in the network. To do that, we had to turn to the natural world. It turns out that throughout all the kingdoms...
There goes that organism again. Let me know what that looks like. I think it looks like a glow sperm. That's what I would call it. But y'all let me know.
Of life in plants and funguses and bacteria and so on, you can find photosynthetic or photosensory molecules that convert light into electricity. So we borrowed these molecules from nature, and then using tricks from the field of gene therapy, we can put them into neurons. Now, these molecules can convert light to electricity, and they do it just in the neurons that we want to control, and not all their neighbors. So we can deliver these molecules to some cells and not others, and then when we shine light on the network, we can turn on or off that subset of the cells. If we can turn on or off a set of cells that's embedded within this dense matrix, we can figure out how do they contribute to a behavior. For example, if we can turn on a set of cells, we can figure out what kinds of behaviors can it initiate. If we can turn off a set of cells, then we can delete it momentarily and figure out what it's necessary for. So by being able to dial in information into cells in the brain and to delete them, we can try to figure out how they contribute to networks and the behaviors and diseases that arise from brain computations. We can hunt down the exact set of cells that are contributing to a specific disease state, or which, when activated or shut down, will remedy that disease state. That's really important because right now, a lot of drugs are developed that target molecules, but molecules are found throughout the brain. And in fact, many cells in the brain might be very molecularly similar to one another. If we can target circuits in the brain, though, we might be able to develop much more specific drugs. Imagine if we could hunt down the exact set of cells in the brain that, when activated, remedy a brain disorder, and then we can go in and look at the exact molecules in those cells. Maybe we can find drug targets that are much more specific than existing ones. You might also imagine that we could use optogenetics to directly control brain circuits.
So for me, what it sounds like he's saying is we're using this type of optogenetics to learn how we can use and make and create medicines that activate certain regions of the brains responsible for certain behaviors or patterns, or whether it's the visual cortex for like seeing, or other parts of your brain that are used for decision making. We can like specifically target these circuits.
Patients with brain disorders. Electricity is used to stimulate the brain in deep brain stimulation. If instead we could actually aim light at certain cells and turn them on or off, we might be much more specific. Rather than using electricity to turn on and off the cells of the brain and have many kinds of cells activated, the ones we want to affect as well as all their neighbors, if we could make just one disease-associated subset associated with light and we can turn them on or off, we might be able to treat them with much more specificity. So far, optogenetics has had a lot of impact in the scientific world, but it hasn't been used in any human patients yet. There are a couple reasons why. One is that it requires a gene therapy to deliver the gene that encodes for these light-activated molecules into the body. Currently in the US, there are no FDA-approved gene therapies. In Europe, there's just one. Another
gene therapies, I think that's what Kylie Jenner got to modify her baby's face. Am I right or wrong, chat, or am I making that up?
issue is that these molecules come from organisms like algae and bacteria. And so if we are putting these molecules into the body, would they be detected as foreign agents and attacked by the immune system, for example? What we need is a paradigm shift in how we think about treating brain disorders. And one of our major stances is that we need new technologies if we really want to either understand the principles of treating brain disorders.
Sorry to Kylie Jenner if I'm making that up, but I mean, from what I heard, they're already doing gene therapy and editing their child's DNA when it's already in the wound. I think it's a very expensive process, but look that up.
Hunting down the exact cells in the brain that can help us treat brain disorders, or to develop new modalities, new forms of energy, new strategies for treating brain disorders by correcting the computations within the brain.
Wow. All right. A little video from MIT there. Got another article that I want to go into. Our next topic here, we're going to be talking about T cells. I haven't read any of these articles, by the way, so I'm reading them all with you guys here for the first time as we bring them up. So I'm learning with you guys, which is always fun. It's always fun to learn new things. Expand your knowledge, exactly. T cells may be living anti-aging elixir. This was posted in 2024 of January. Researchers found that T cells can be genetically reprogrammed to target and eliminate senescent cells, which are like cells that have died or are no longer active, which contribute to aging-related diseases. By using CAR or chimeric antigen receptor T cells in mice, they achieved significant health improvements, including lower body weight, enhanced metabolism, and increased physical activity. This groundbreaking approach, offering long-term effects from a single treatment, could revolutionize treatments for age-related conditions like obesity and diabetes, transcending the potential of CAR T cells beyond the current use in cancer therapy. Reprogrammed CAR T cells can effectively target senescent cells, reducing age-related health issues. In mice, a single treatment with these modified T cells offer long-term health conditions, a major advantage over current therapies requiring repeated doses. This discovery expands the medical applications of CAR T cells, previously known for treating blood cancers, to potentially combating aging-related diseases. The fountain of youth has eluded explorers for ages. It turns out the magic anti-aging elixir might have been inside us all along. Cold Spring Harbor Laboratory assistant professor Karina Amor Vegas and colleagues have discovered that T cells can be reprogramming to fight aging, so to speak. Given the right set of genetic modifications, these white blood cells can attack another group of cells known as senescent cells. These cells are thought to be responsible for many of the diseases we grapple with later in life. Senescent cells are those that stop replicating. As we age, they build up in our bodies, resulting in harmful inflammation. While several drugs currently exist that can eliminate these cells, many must be taken repeatedly over time. As an alternative, Amor Vegas and colleagues turned to a living drug called CAR, or chimeric antigen receptor T cells, that they discovered CAR T cells could be manipulated to eliminate senescent cells in mice. As a result, the mice ended up living healthier lives. They had lower body weight, improved metabolism and glucose tolerance, and increased physical activity. All benefits came with any tissue damage or toxicity. So if we give it to aged mice, they rejuvenate. If we give it to young mice, they age slower. The other therapy right now can do this, says Amor Vegas. Perhaps the greater power of CAR T cells is their longevity. The teams found that just one dose at a young age can have lifelong effects. The single treatment can protect against conditions that commonly occur later in life, like obesity and diabetes. T cells, another thing that can combat against obesity and diabetes is exercise, diet. Um, but yeah, that's another story, right? T cells have the ability to develop memory and persist in your body for really long periods, which is very different from chemical drug, explains Amor Vegas. With CAR T cells, you have the potential of getting this one treatment, and then that's it. For chronic pathologies, that's a huge advantage. Think about patients who need treatment multiple times per day versus you get an infusion, and then you're good to go for multiple years. CAR T cells have been used to treat a variety of blood cancers, receiving FDA approval for this purpose in 2017. But Amor Vegas is one of the first scientists to show that CAR T cells' medical potential goes even further than cancer. Amor Vegas lab is now investing whether CAR T cells let mice live not only healthier but also longer. If so, society will be one mouse step closer to the coveted fountain of youth. All right, so this is research about aging and genetics. CAR T cells. Let's see how these work. CAR T cell therapy. Share. Got a video from YouTube. So right into it. CAR T cell therapy, leukemia and lymphoma society. This was posted 4 years ago. We are currently in 2024. So that would make this 2020.
At LLS, we always say that beating cancer is in our blood. It's a phrase that captures the heart and soul of what we do. But what if your own blood could actually be turned into a meaner, leaner cancer-beating machine? Well, that's what doctors and researchers are doing right now with something called chimeric antigen receptor T cell, or CAR T cell therapy. To understand it, we need to start with T cells, a type of white blood cell that patrols our bodies for signs of disease. See, all cells have protein fragments called antigens on their surface. Some antigens are tumor-specific, meaning they're specific to certain cancers. And it is these tumor-specific antigens that ultimately alert your body's immune system, specifically your T cells. See, T cells have receptors on their surface that are uniquely designed to latch on to antigens of cancer cells. They then kill the harmful cells by injecting them with toxins. It's a pretty amazing system when it works. The trouble is, cancer is smart. Cancer cells can disguise themselves so our T cells don't recognize them as foreign. And when that happens, the cancer grows, leaving one of your body's most powerful defenses, well, kind of powerless.
You heard that? We got some sneaky cancer cells. They can disguise themselves as other cells so your T cells can't kill them. That's literally warfare at the biological level got going on here.
So researchers asked themselves, what if we could reprogram a person's own T cells to break through cancer's defenses? And that's what CAR T cell therapy is all about. In a nutshell, the doctor takes out some of your own T cells and sends them to a lab. The laboratory technicians turns them into super cells, CAR T cells. They do this by modifying the new CAR T cell receptors to recognize your specific cancer and make those receptors, well, more receptive to its antigens. The new cells are reproduced a few million times and then frozen so doctors can begin treatment when the time is best for you. Before you're able to get your new CAR T cells, you'll be given some.
It's a lot like stem cells, how they're extracted from your blood and bone marrow and then infused and then frozen until you get them, or from the placenta frozen, the umbilical cord and frozen and then given to the patient, so to save them or whatnot, the health of the cells. So if they tell you that cancer is not healable or treatable, just know that there are therapies out there that are targeted for cancer. So don't always believe in industrial healthcare system. But that's another controversial topic that don't really want to dive too much into, but just do your research,
Thing called lymphodepleting chemotherapy, which will make room for your new supercells. Then, when it's time, your doctor will introduce the new and improved T cells back into your body. The good news is that they start attacking the harmful cells right away. The bad news is that the therapy has a high likelihood of causing side effects and making you sick. Many patients experience symptoms typical to a bad case of the flu. Patients also report nausea, dizziness, and loss of balance, among other symptoms. And make no mistake, in some rare cases, patients can experience more serious side effects, like confusion, seizures, and even stroke-like symptoms. Your doctors will want to monitor your condition, so they'll likely require you to stay near a hospital or perhaps even check into one. But for many patients, these side effects are temporary and can be managed with
You're over here making all type of noise during this podcast.
medications or other
Quiet on set.
procedures. What's more, researchers are making great strides in controlling and reducing those side effects. So what happens after the sickness passes? Well, this is the really good part. These stronger and faster CAR T cells just keep reproducing in your body for a long time after the infusion, continuing to fight the cancer if it returns. And in many cases, they continue to work long after a patient enters remission. Right now, this therapy is typically only given to patients with specific types of blood cancer who have not responded to common cancer treatments. And only your doctor can tell you if this therapy is a real option for you. But while the results are early, they're incredibly promising. And encouraging studies for other types of blood cancers are moving forward at great speed. Yes, beating cancer is in our blood, but the key to beating your cancer might just be in yours.
Interesting. Using your own blood to heal your own cancer. Who would have thought of it, right? So yeah, not many people do know about T-cell therapy, but that is a treatment. I mean, it's killing cancer cells, which are senescent cells or dead cells that have pretty much gone rogue in your body and do damage. So we got another video here talking about, we've been going for 37 minutes here. I do want to get into about 2 more topics here. So we'll skim through this one. We're going to get into fasting, a little bit of fasting research coming out that's been out in some sleep study research, and might get into some nicotine type stuff. So let's jump into it. This video is another YouTube video. Shout out to Orlando in the chat. He says, I love lamp. I'm guessing he's talking about this Stormy lights in the back, and they're pretty nice. I like them too. This video is from 5 years ago, posted by Dr. Susan Love Foundation. Chimeric antigen receptor T cell or CAR T therapy.
In 2017, the Federal Drug Administration approved the first ever gene immunotherapy to treat cancer in the U.S. It's called Chimeric Antigen Receptor T-cell therapy, or CAR T for short. This personalized therapy has shown remarkable results treating some children and young adults with acute lymphoblastic leukemia, a type of blood cancer. A few months later, a similar treatment was approved for adults with certain types of non-Hodgkin lymphoma.
Gene therapy, a new and promising area of cancer treatment, is now being studied to learn whether or not it'll be effective in breast cancer patients.
CAR T uses our body's natural ability to detect specific proteins that help them identify and tag cancer cells.
By tweaking the T cells so that they recognize specific proteins, researchers have been able to train a patient's own immune system to identify and eliminate their cancer cells.
Here's how it works. A doctor will take some blood from the patient and isolate the T cells from the rest of the fluid. In a lab, these cells are modified to look for and kill the patient's cancer cells. The newly armed T cells are then put back into the patient's blood, where they go to work knocking out the bad cells. It is important to note that CAR T therapy is given only once, and it is expensive. One treatment is priced at $373,000, the other at $475,000. As with most cancer treatments, there can be side effects. Some of these can be life-threatening. Doctors are learning how to manage these side effects, but it's still not fully understood why they occur.
It's not yet clear if these treatments will work for breast cancer patients.
Additionally, CAR-T doesn't work for everyone, but there is no question that it is a significant advance in how we treat cancer. Currently, a small number of trials are studying CAR-T therapy in metastatic breast cancer, and more are expected soon. Immunotherapy is a very exciting field, with many new techniques like CAR-T being developed. These kinds of breakthroughs can give hope to many cancer patients.
CAR-T healing are being used to heal, and treat all type of different cancers, including breast cancer. So yeah, guys, there's options out there if you know anybody who has cancer, right? There's hope. Not a lot of people have to go through that, and hopefully you don't in your life, but it can be a very challenging and scary situation, I would imagine, to be diagnosed with any type of cancer. And it's possible for all of us as we age, because we are all of our cells are like rapidly dying, right? And so yeah, that's what this next topic is going to be teaching us about, and how to prevent that inflammation, because it's going to be talking to us about fasting, which can reduce inflammation. So researchers uncovered a new mechanism by which fasting reduces inflammation, a key factor in chronic diseases. Their study reveals that fasting increases blood levels of arachidonic acid, which inhibits the LRP3 inflammasome, thereby reducing inflammation. This discovery sheds light on the anti-inflammatory effects of fasting and offers insights into the benefits of calorie restriction for conditions like obesity, diabetes, heart disease, and neurodegenerative disorders. So anything affected by the central nervous system. You can also experience benefits such as a spinal cord injury, right? The researcher also provides clues to how drugs like aspirin might function, further highlighting the intricate relationship between diet, inflammation, and disease prevention. Fasting elevates arachidonic acid levels in the blood, leading to reduced activity of the NLRP3 inflammasome and inflammation. The findings offer a potential explanation for how fasting and calorie restriction can protect against chronic inflammation-related diseases. This research may often explain the anti-inflammatory effects of non-steroidal anti-inflammatory drugs like aspirin, which increase arachidonic acid levels. So taking aspirin can kind of mimic the effects of fasting, guys. And also, we all know that if you have ever fasting is not something that is easily done. You get used to your diet, your lifestyle, and take the discipline. And you got to do it the right way. You can't just think that just not eating is good. You also need to be well hydrated. There's a lot of processes, and there's different types of fasts. You have dry fast, where you don't eat or drink anything. I think that's probably one of the most difficult things you could probably do to yourself. And then you got fasting with water, or water fasting. And then you got the intermittent fasting, where you eat throughout a certain window of the day, typically like 4 to 8 hours windows is what people have. So they'll eat like 2 meals in that time frame, or 1 meal a day. You also have that. So there's a lot of different ways. That would be like a 24-hour fast. And then you got the alternate day fasting as well, eating every other day. And then you got just long fast. So there's different ways of accomplishing the goal, but can have a lot of benefits, as we're going to read here. Cambridge's scientists may have discovered a new way that fasting reduces inflammation, a potentially damaging side effect of the body's immune system that underlies a number of chronic diseases. In a paper titled Arachidonic Acid Inhibition of the NLRP3 Inflammasome is a mechanism to explain the anti-inflammatory effects of fasting. Published in Cell Reports, the team describes how fasting raises levels of chemical in the blood known as arachidonic acid, which inhibits inflammation. The researchers say it may also explain some of the beneficial effects of drugs like aspirin. Scientists have known that for some time that our diet, particularly a high-calorie Western diet, can increase our risk of diseases, including obesity, type 2 diabetes, and heart disease, which are linked to chronic inflammation in the body. So as we scroll down here, continues by saying inflammation is our body's natural response to injury or infection, but this process can be triggered by other mechanisms, including by the so-called inflammasome, which acts like an alarm with our body cells, triggering inflammation to help protect our body when it senses damage. But the inflammasome can trigger inflammation in unintentional ways. One of its functions is to destroy unwanted cells, which can result in the release of cells' contents in the body, where they trigger inflammation. So these, like using a anti-inflammatory, they're saying can trigger inflammation by releasing inflammasomes in the body, triggering more inflammation. Professor Claire Bryant from the Department of Medicine at the University of Cambridge said, "We're very interested in trying to understand the causes of chronic inflammation in the context of many human diseases, and in particular the role of the inflammasome." What's become more apparent over recent years is that the inflammasome, in particular the NLRP3 inflammasome, is very important in a number of major diseases such as obesity, atherosclerosis, but also in diseases like Alzheimer's and Parkinson's disease, many of the diseases of older age people, particularly in the Western world. It's a long article. Fasting can help reduce inflammation, but the reason why has not been clear. To help answer this question, a team led by Professor Bryant and his colleagues at the University of Cambridge and National Institute for Health in the U.S. studied blood samples from a group of 21 volunteers who ate a 500-calorie meal and then fasted for 24 hours before consuming a second 500-calorie meal. The team found that restricting calorie intake increased levels of a lipid known as arachidonic acid. Lipids are molecules that play important roles in our body, such as storing energy and transmitting information between cells. As soon as individuals ate a meal again, levels of arachidonic acid dropped. When the researchers studied arachidonic acid's effect in immune cells cultured in the lab, they found that it turns down the activity of NLRP3 inflammasome. That surprised the team, as arachidonic acid was previously thought to be linked with increased levels of inflammation, not decreased levels. Professor Bryant, a fellow of Queen's College, Cambridge, added, "This provides a potential explanation for how changing our diet, in particular by fasting, protects us from inflammation, especially the damaging form that underpins many diseases related to a Western high-calorie diet. It's too early to say whether fasting protects against diseases like Alzheimer's and Parkinson's disease, as the effects of arachidonic acid are only short-lived, but our work adds to a growing amount of scientific literature that points to the health benefits of calorie restriction. It suggests that regular fasting over a long period of time could help reduce the chronic inflammation we associate with these conditions. It's entirely an attractive idea." So they're saying they're unaware or they're not completely positive about fasting protecting against Alzheimer's and Parkinson's, but it creates the environment that reduces the inflammation that cause these conditions. The findings also hint that one mechanism whereby a high-calorie diet might increase the risk of these diseases. Studies have shown that some patients have had a high-fat diet have increased levels of inflammasome activity. There could be a yin and yang effect going on here, whereby too much of the wrong thing is increasing our inflammasome activity, and too little is decreasing it, said Prior Bryant. Arachidonic acid could be one way this is happening. The researchers say that the discovery may also offer clues to an unexpected way in which so-called non-steroidal anti-inflammatory drugs such as aspirin work. Normally, arachidonic acid is rapidly broken down in the body, but aspirin stops the process, which can lead to an increase in levels of arachidonic acid, which can reduce inflammasome activity and hence inflammation. So Professor Bryant said it's important to stress that aspirin should not be taken to reduce risk of long-term diseases without medical guidance, as it can have side effects such as stomach bleeds if taken over a long period. Personally, I have never been one to take aspirin or Tylenol if I have a headache. I usually just drink some water and thug it out. Usually there's other, underlying reasons that you may have a headache, or, I guess you can also use it for anti-inflammatory reasons, like we just read. But, it comes with side effects, as we just read as well, such as stomach bleeding. So who wants to be taking that crap with issues like your stomach bleeding, right? This is from Lex Friedman, a clip where he talks with Andrew Huberman about fasting. Posted, this was 3 years ago. So shout out to Lex Friedman and Andrew Huberman, two podcasters I listen to a lot. Friends with Joe Rogan down there in the Austin area. A lot of stuff going on there, down there in Texas.
I mean, you also mentioned fasting potentially through this 2-day thing.
Elon Musk as well. I believe Elon has Tesla and a home in, like Tesla factories and a home in out there in Austin as well.
It'd be cool to get your thoughts about fasting in general. Do you think on a personal level and at a higher sort of level of studies that you're aware of and physiology and so on, what do you think about intermittent fasting, of like not eating for 16 hours and then having an 8-hour window, or something I've been doing a lot recently, which is eating only once a day? So that's 24-hour fast, I guess, one meal a day, or something I've been thinking about doing, haven't done yet, of doing like 72 hours, or some people do like 5-day fasts in general. So this would be, for this particular run, would be a 48-hour fast if I don't eat at all.
Let me know, all the OGs in the chat, who have seen my 3-day fast. Well, I only drunk water and ate shrooms. It's still up on my YouTube, surprisingly, called I Only Ate Mushrooms, or I Only Ate Shrooms for 3 days. Go check that out if you haven't yet. Pretty interesting video.
What do you think about that for
YouTube.com/StormyFriends.
performance, for mood, for all those kinds of things?
I can speak a little bit to the science and a little bit of my own experience, and then some anecdotes of people that have done very hard, very long duration things and what they've told me. So I just want to make sure I'm separating those out so people know my sourcing. I think now none of this is about the actual long-term nutritional benefits of one thing or the other. But if you look at the science on intermittent fasting, it's pretty remarkable. Before I was at Stanford, my lab was in San Diego. One of my colleagues was Sachin Panda at the Salk. He's a phenomenal biologist and researcher, wrote a book called The Circadian Code. It's very, very good, and kind of popularized intermittent fasting, although there were others that had talked about this before. Ori Hofmeckler talked about the warrior diet. People probably might not know who Ori is, but he's sort of the originator of this business of intermittent fasting, eating once a day or limited. Anyway, Sachin has published papers, peer-reviewed papers in very good journals like Cell and elsewhere, showing that limiting the consumption of calories to 8, 4, 6, or 8, or even 10 hours of every 24-hour cycle, and keeping that more or less correlated with the light, with when the sun is out, leads to less liver disease, improved metabolic markers, less body fat, et cetera. In the mouse studies, they even gave the mice the choice to eat whatever they wanted, as much as they wanted, as long as they restricted it to a certain period within the 24-hour cycle. They did great. They maintained a healthy weight or even lost weight when they took the same amount of food.
I mean, if you've ever done a fast, especially an extended fast, like, that they can feel great, but I mean, it's all about doing it right too, right? And the refeeding process apparently is like one of the most important parts, what you put into your body after, all that fasting and the benefits from that. It's multi-step process.
And they stretched it out across the entire 24-hour cycle. So this is eating every hour or 2 hours. The animals got fat and sick. So it's pretty remarkable data. How much of that translates to humans isn't clear, but one thing that's really clear with humans is adherence, right? We could talk a lot about nutrition, and some of the problems with the studies on nutrition is that what people will do in a laboratory is often hard to do in the real world. Low carbohydrate diets, because they tend to focus on foods that have high amino acid content, like meats. Generally, people are less hungry on those than they are on calorie matched diets of fruits and vegetables and carbohydrates, because when the insulin goes up, you get hungry and you want to eat more. So this is not a push for carnivore or a push against one thing or the other. It's just, there are a lot of factors. But we know for sure that when you're fasted or when you have low amounts of carbohydrate in your system, complex carbohydrate, your alertness is going to go up. Fast increases alertness.
That's big facts. If you've ever done a fast, you realize that it's so good for like your brain and decision making. Like after you feel so clear, and I feel like it's so much easier to make decisions, like way less brain fog. I guess that has to do a lot with the inflammation that it helps with.
And epinephrine for the sole purpose of getting you to go out and find food. Can you imagine if our ancestors got hungry and they were like, oh, I'm too tired to go find food? We wouldn't be here. It'd be like robots or something. One of your alien buddies will be like running the planet. Dream. So I think that if you want to be alert, fasting or keeping complex carbohydrates to a minimum is very valuable. If you want to sleep and you want to be sleepy, ingesting foods that have a lot of tryptophan, which is the precursor to serotonin, so complex carbohydrates like rice and grains, turkey, white meats, those things do create a sense of sleepiness. However, there is a caveat, and this is one problem with the once a day meal, is that anytime you have a lot of food in the gut, you're increasing sleepiness because you're diverting blood to the gut. It's going to trigger the vagus to signal to the brain to shut down your system and utilize those nutrients, digest and utilize those nutrients. So I've done the once a day eating thing. The problem is I eat so much in that meal that I'm exhausted. And so it doesn't always lend itself well to the schedule. But so in a 6 or 8 hour eating block for me is a little bit better. Mm-hmm. I do eat carbohydrates. I'm probably one of the few people left on the West Coast that actually consumes carbohydrates, and will say that out loud.
I don't know people eat carbs anymore. That's weird.
They don't.
Where do you even find carbs?
What do you find? Yeah, I like oatmeal. I like rice. The other time is if people are doing very high intensity weight training, they need to replenish glycogen.
Yeah. On the alertness side, I do feel like it's probably person dependent.
I mean, I do feel that in general when I eat carbs, I feel a lot slower and fuller and stuff like that. And when I tend to eat more just pure protein meals, I feel great. Like recently I've been eating like lamb roast that I've been making in the crock pot, and just been eating that with like some eggs, just a pure protein packed meal, no carbs, no rice, no vegetables, nothing, just straight meat, eggs, stuff like that. I feel great. So carnivore works for me personally, I think.
For me, alertness, being alert makes my life better in a lot of ways, more than just the alertness itself. Like, for example, one of the things I discovered with fasting is that when I was training twice a day in jiu-jitsu, for example, and competing and so on, I performed way better at things that you traditionally would say you need carbs for, which is explosive movements and all that. I don't know if I actually perform better in terms of like the force of the explosion, the explosiveness. What I do know is the alertness resulted in me doing the technique more precisely.
That's the dopamine and epinephrine system in action. And there's there are some other just pure
go back to that decision-making process. I mean, it may not, obviously you're not taking in calories, so you're not going to be gaining muscle mass and size and power, but it will eliminate the inflammation you have built up from doing exercises and allow you to perform better.
Truly physical aspects to one diet versus the other that can be complicated. If you're ingesting carbohydrates, complex carbohydrates, you're going to replenish glycogen, which is great, but they also tend to be bulky and fibrous. And I've never rolled jiu-jitsu, but running when you have a lot of bulky fibrous food in your gut or in your intestine, it can be a barrier, it can be uncomfortable. And so some people do really well on low carbohydrate, meat-rich diets because they're just not as bloated. They're not carrying as much water and other stuff. Carbohydrate carries a lot of water molecules with it. So there are aspects to being able to train and being really explosive because you feel light. One anecdote that really, again, I'm not encouraging any one particular kind of diet, but I have a friend who was in the in the SEAL teams, I happen to know a number of people in that community, and he told me that he did this very long fast. It was a fast that I think you get to eat a little bit of soup or broth, and there's like a bar or something, but it's like a 9-day thing.
Mm-hmm.
And he's a very strong athlete, and he said that on day 6 or 7, he was running up some hills or something while he was on deployment, and he felt amazing. He kind of hit this other level. He was somebody who had boxed in the Naval Academy. He was somebody who knows and knew high output, and he felt like he discovered the 13th floor, that there was another floor to this performance space that he hadn't experienced except while he had fasted. And he said that was a remarkable clarity of mind, energy. It's a little bit of what you described.
Mm-hmm.
He described a kind of suppleness and explosiveness. So there's probably something there.
On which day?
Once he was in the 5th or 6th day of the fast.
See, this is the thing, is I've never been there on the 2nd, 3rd, 4th, 5th day, that kind of thing. But
Yeah,
when I just don't
I think my longest is 72 hours, and right after I probably didn't have to eat. I could have went longer, but I mean, I was just really craving some, you just get used to eating, and you just get used to that mouth satisfaction, But I felt amazing. Like I said, I did video and talk about it on my YouTube. 72 hours, only eating mushrooms. Go find that.
don't eat for 20 hours. Many times through my training, the clarity, it's like you feel like everyone is moving super slowly,
mm-hmm.
and you're able to like dominate people you weren't able to before. It's like,
well, you might have slipped into or switched over rather into full ketosis.
So yeah, the ketogenic diet. A lot of people are familiar with that. It's a one of those diets that's probably good to try for most people. Of course, do everything slowly, and get into to where you can maintain the diet, because it's a complete lifestyle change with how you're eating and whatnot. But a lot of people see benefits with that type of diet because it reduces carbs, and they feel a lot better and less heavy and bloated all the time. But for some people, apparently carbs don't have the same effect. They can eat a little bit of carbs and still maintain proper function and feeling, and they feel great. So I don't know. It's obviously person dependent. So this next topic we're going to be jumping into here, titled Sleep's Role in Brain Health. So a little bit of sleep studies here. I always find sleep topics and articles like in news and new studies that are coming out interesting. So let's get into it. Posted on the 30th of January 2024. New study reveals how sleep duration impacts brain health, specifically relating to stroke and dementia risks. Analyzing brain images of nearly 40,000 middle-aged participants, the study found that both short and long sleep durations are associated with negative changes in brain structure. So both too little sleep and too long of sleep durations can negatively affect you guys. So I guess there's a sweet spot of how much sleep you're supposed to get. These changes include higher presence and volume of white matter, hyperintensitities. Higher presence of and volume of white matter hyperintensities, or WMH, and reduced fractional anisotropy, and are indicators of brain aging and dementia risk. The researcher underscores sleep as a key factor in maintaining brain health and highlights middle age as a critical period of sleep adjustments. As you get older, guys, you realize that sleeping is just so much more important. And like me personally, I need to get a certain amount of sleep, or I just don't function the same the next day. At this point, I used to be able to go to sleep at 3, 4, 5 o'clock in the morning, wake up at 8 o'clock and go to school, but if I did that nowadays, I feel like I just would not properly function. I would not be able to make decisive decisions, or having a podcast like this would be impossible. So yeah, sleep's important to me. But it wasn't always this way. Inadequate sleep, both too little and too much, is linked to increased WMH or white matter hyperintensities, larger WMH volume, and lower fractional aneosarchy. These brain changes are associated with higher risk of stroke and dementia. The study emphasizes the importance of optimal sleep between 7 and 9 hours for brain health in middle-aged individuals. And I know the common term is usually always get at least 8 hours of sleep, but staying between 7 and 9. So if you get in that range, you're doing good for yourself. Getting either too much or too little sleep is associated with changes in the brain that have been shown to increase the risk of stroke and dementia later in life. A recent study finds the research is published in the Journal of American Heart Association. Conditions like stroke or dementia are the end stage result of a long process that ends tragically, says Santiago Clacchiati, Tuozo, a medical doctor, T23 postdoctoral fellow in the Falcon Lab at Yale School of Medicine and first author of the study. We want to learn more how to prevent these processes before they happen. In one of the largest neuroimaging studies of its kind to date, the Yale team examined brain images of close to 40,000 healthy, middle-aged participants to evaluate how sleeping habits might impact two measures of brain health: white matter hyperintensities, which are lesions of the brain indicating brain aging, and fractional aneosarchy, which measures the uniformity of water diffusion along nerve axons. More WMH, larger WMH, and lower fractional and aneosarchy are associated with the increased risk of stroke and dementia. Researchers found that compared with optimal sleep, between 7 and 9 hours per night, participants with short sleep had higher risk of WMH presence, larger WMH volume where WMH was present, and lower fractional aneosarchy. Long sleep, averaging more than 9 hours per night, was associated with lower fractional aneosarchy and with larger WMH volume, but not with risk of WMH presence. These findings add to the mounting evidence that sleep is a prime pillar of brain health, says Clocchiati Tuozo. It also provides evidence toward helping us understand how sleep and sleep duration can be modifiable risk factor for brain health later in life. Researchers say the study highlights middle age as an important time to adjust our sleeping habits to support brain health. Sleep is starting to become a trending topic, Clocchiati Tuozo says. We hope this study and others can offer insight into how we can modify sleep in patients to improve brain health in years to come. So we scroll down here. Cyprien Riviere, Daniela. Okay, so these are the people that were co-authors of the study. So as we know, guys, a lot of people already know this, but sleep plays a huge role in our brain health. So as we sleep, right, we're going into a process where brain is in repair mode and is healing. Apparently, sleep, I find so interesting how we just, close our eyes and give it some time, and all of a sudden you're in a completely different dimension, right? So like, how interesting is it that we just need to do this process every day in order to function properly? Like, it's something that if you did not do, you would die. You basically wouldn't function. And so it's important to a lot of you listening out there who like to stay up all night and don't like to, prioritize your sleep. It's really harming you, it's really important to get yourself a consistent sleep schedule. Although, not easy to do. It takes discipline, but if you can do that, you're benefiting yourself.
So that's referring to more that there should be a consistent habit
Should you get 8 hours of sleep? Another clip from Lex Friedman and Andrew Huberman.
Versus the total amount. To me, like the entirety of the picture of sleep is similar to nutrition in that it feels like there's so many variables involved and it's so person specific. So, a lot of studies, I mean, this is the way of science, has to look in aggregate the effects on sleep. It doesn't focus on high performers, which are individuals ultimately. Like the question isn't, so it's a very important question, is like what kind of diet fights obesity, reduces obesity. It's another question, what kind of diet allows David Goggins to be the best version of himself? So these high performers in different avenues. And the same thing with sleep. Like people that tell me that I should get 8 hours of sleep, it's like, I get it, and they may be right, but they may be very wrong. And
there's no evidence that 8 is better than 6. That you could very well do better on 6 than on 8. There are a few other things that turn out to be strong parameters for success in this domain. For instance,
but apparently, that's contradictory to what we just read. That article saying that the Goldilocks zone would be around 8, 7, between 7 and 9. He's saying that 6 is no better than 8, and we just read that that's not true. So, Huberman, challenging you.
Your entire life, waking or asleep, is broken up into these 90-minute ultradian cycles. If you look at ability to attend,
not really. Shout out to Andrew Huberman. He knows exactly what he's talking about, but he may be wrong here.
Or do math problems or do anything, drive, performance tends to ramp up slowly within a 90-minute cycle, peak, and then come down at the end of that 90-minute cycle. And in sleep, we go through these stage 1, 2, 3, 4, REM, etc. We'll talk more about that if you like. Those on 90-minute ultradian cycles as well. Ending your sleep after a 90-minute cycle, at the near the end of a 90-minute cycle, say at the end of 6 hours, in many cases is better for you than sleeping an additional hour, 7 hours, and waking up in the middle of an ultradian cycle. And there are a few apps that can measure this based on body movement.
Okay, that actually makes more sense. He's talking about the different cycles we go through during sleep. You don't want to interrupt the cycle while it's in process, I'm guessing. So 6 hours would be optimal versus 7 when you're in the middle of a
and things like that have you
sleep pattern or cycle or whatever.
Your alarm go off at the end of an ultradian cycle. And if you wake up in the middle of an ultradian cycle, sometimes, not always, you can be very groggy for a long period of time. I certainly do better on 6 hours than I do on 7. I happen to like an 8-hour sleep. It feels great, but I haven't slept an entire 8 hours without waking up in the middle of the night at some point in, I don't know, forever. I can't remember. It's probably some point in infancy, but.
All right. Fire. Shout out to Huberman and Lex Friedman. Well, my screen's going crazy here. So yeah, we got another YouTube video. This is with Huberman again, but he's talking to Dr. Gina Poe, and it's how to fall asleep. That may be the most difficult process of the sleep process, or the most difficult step, right? Is to actually get yourself to be able to relax enough and get into the zone to be able to fall asleep. A lot of people got different lives and a lot of different things, and depending on how your lifestyle is and situation you got going on in your home, will determine on when you're able to get to sleep and how you're able to fall asleep. But so yeah, let's get right into this video. Clip posted 8 months ago, so this is a lot more recent.
I'm trying to get into calmer states prior to sleep, and some ways to do that. I'm a big fan, and I've talked a lot before on this podcast about things like yoga nidra, which is a non-movement based practice, sometimes called non-sleep deep rest, where people actually take some time each day to practice how to go into a more parasympathetic, aka relaxed state deliberately, because it's a bit of a skill. Yeah, it's and there's some good data, really, mostly out of a laboratory in Scandinavia, showing huge increases in nigrostriatal dopamine when people go basically engage in a practice of deliberate non-movement, and that the brain actually enters states of a very shallow sleep. So it's sort of nap-ish, but the idea is to actually stay awake but motionless. And it does seem to restore a certain number of features of neurochemistry, but perhaps more importantly, it teaches people to relax, which is something that most people are not very good at. But in any event, and people who listen to this podcast have heard me say this over and over again, so I sound like a broken record, but this practice as a zero-cost practice that doesn't require any pharmacology does seem to really enhance people's ability to fall asleep more quickly and to fall back asleep if they wake up in the middle of the night. So in any event, another plug for NSDR.
Yoga Nidra. How many of y'all have done yoga? I mean, it is very relaxing. I remember doing yoga, especially during football. We used to do like group yoga sessions, and we used to have a instructor come out and get us through the, you know, different positions and poses, the downward dogs, you know, all the different type of crazy positions. And I consider myself like very flexible, very athletic, especially, you know, during those times. And I used to love doing yoga. It's very relaxing, and it's actually a lot harder than you think, you know, to hold those positions, and it takes a lot of core strength.
Our Yoga Nidra.
Well, I just also want to add to that. That's one of the reasons why insomnia is so insidious, is because when people feel like they haven't gotten enough sleep and aren't getting enough sleep, and they become anxious about getting enough sleep, and then you're anxious before going to sleep, like, I'm not going to fall asleep, it's going to be 45 minutes in, and then that's a positive feedback loop. So you need to break that loop, say, okay, my body's going to get as much sleep as it needs, I needn't worry about it, and then practice this relaxation to say, hey, it's all okay, it's going to be all right, and then concentrate on things that relax you, whether it's concentrating or not concentrating, whatever it is. You mentioned Yoga Nidra, and that reminded me of transcendental meditation, which is something that also hasn't been studied well, largely because we can't ask non-human animals to do it. And so we don't know what's happening with our neurochemistry and our brain activity in a deep and meaningful way. But one thing that has been shown in those that can do it really well is that theta activity that I said happens when you're learning something or when you're in REM sleep. It's well established and increases during the transcendental meditation. So it might be that some states of meditation could in some ways replace or mimic some functions of, for example, REM sleep. But again, we don't know if all the neurochemistry is right to do, for example, the thing that I was talking about, which is erasing the novelty encoding structures of the brain. That needs an absence of norepinephrine and serotonin, which we don't know if that goes away with transcendental meditation. We just don't know the answer to that yet.
Yeah, the studies on Yoga Nidra and sleep replacement are kind of interesting.
Talking about like getting into these yoga, you know, these transcending within meditation, you can get your body to produce the hormones that are associated with sleep and get those brain areas fired. So it can replace, you know, sleeping yoga. You know, so that's interesting.
It does seem to be the case that nothing can really replace sleep except sleep, but that if one is sleep deprived or is having trouble falling back asleep, that these things like, and I acknowledge this is essentially like Yoga Nidra, but we now call it non-sleep deep rest or NSDR, because oftentimes for names like Yoga Nidra act as a kind of a barrier for what would otherwise be people willing to try a practice. Right. It sounds mystical.
And yoga,
it sounds like flying carpets and woo, you know, it sounds like you have to go to Esalen. By the way, Esalen's a beautiful place, but it sounds like you have to go there or live in the West Coast to believe in this stuff, but it's simply not the case. These are practices that are really just self-directed relaxation as a practice that allows people to get better and better at directing their brain states towards more relaxation. Right. And most people have an asymmetry. Like, for instance, most people can force themselves to stay up later, but they have a hard time going to sleep earlier. And that just speaks to the asymmetry that's probably adaptive and survival based, that we can ramp ourselves up far more easily than we can tend to calm ourselves down.
Yeah. And actually, you know, to appeal to other Christians like me, prayer can be a wonderful way to calm yourself down, because through prayer, you're giving your cares to God and saying, you know, and then you are more relaxed. And I just want to say that because the same reason that yoga might put some people off, it might put some people off to talk about prayer.
How many of you pray, Chad? I feel like prayer is very important for every person to do. A little bit of gratitude, a little bit of thanking God, you know, every day, just a little bit, talking to them, building that relationship, continuing that relationship with them, you know. And, you know, it's helped me out a lot. So try to get it in a couple times a day, you know, maybe when I wake up or when I go to sleep, just say a little prayer before I eat something, you know, try to remember to say my prayers.
But it's the same process of being able to relax. And
and get outside our own
Get
experience a
outside
little
our
bit.
experience.
Yeah.
Back out, get a worldview that might actually also help us to relax.
Well, you might be surprised at how many clinicians and scientists who've come on this podcast have mentioned things like prayer from various perspectives, Christianity, Judaism, Muslim traditions, and others, that as a parallel to all of these things. And I think what it speaks to is the fact that ultimately the biological architectures that we're all contending with are going to be identical, right?
Exactly.
And so different ways to tap into them and ones that are congruent with people's beliefs, I think are great.
Yeah, because anything non-congruent with your beliefs is also stressful.
Right, and feels forced. And that's why, this idea of calling it non-sleep deep rest in addition to Yoga Nidra was not to detract from the naming or the history around Yoga Nidra, but I was finding that it was a barrier. Likewise, Yoga Nidra tends to include things like intentions, whereas NSDR scripts, and by the way, we will provide links to some NSDR and Yoga Nidra scripts, but NSDR has no intentions. It's simply body scan deep relaxation based. So it's sort of the scientific version of all of this stuff. And actually we study it in the laboratory and some of the brain states that people go into, but that's a discussion for another time.
This is hard. My mother used to tell me when I said, "Well, we could play, I can't go to sleep." She'd say, "Well, start with your toes and relax." So you would clench your muscles around your toes and relax them and do that all the way from your toes all the way to your head. And I don't know where she got this. It might have been her own common sense, or she might have gotten it from.
That's interesting because whenever I go to sleep, I tend to focus a lot on my body as well. And I try to feel every parts of my body. And that actually helps me go to sleep as well. So I'm glad to hear somebody else is doing that out there. It's not just me.
This NPR show is called The Mind Can Keep You Well. She used to listen to, but that's another intentional relaxation that focuses on the body rather than on your own mental processes. But
I do a little bit of work with the military, and there's a method within certain communities of special operations in the U.S. military where if they can't sleep because they're having challenges sleeping, they will deliberately.
Are you a good sleeper?
Let's see. I gotta do no electronics for 30 minutes beforehand.
Like being on the phone, it'll keep you up. Yeah, I'm like that too. Like, yeah, I gotta have it pitch black as well. Like,
deliberately try and relax their facial muscles in particular, like sort of drape the facial muscles and use long or exhale emphasized breathing does seem to increase the probability of transitioning back into sleep. And those are hallmarks of Yoga Nidra, non-sleep deep rest, body scans. And so I think all of these things converge on a common theme. As neurobiologists, we can say all of the things that we are describing certainly move the needle away from locus ceruleus activation. I mean, we haven't done the experiment to really look at that, but it seems all these things are counter to noradrenaline release.
Right. Another one is yawning. Yawning in itself is that kind of sort of tensing of all the muscles in your face and then relaxing them. So it might be why we yawn. We don't know why we yawn yet, but it might also it would be really great. Actually, animals yawn too.
I told we yawn to like relax, or that's the brain telling you it's tired. And
Maybe it hasn't improved.
you write that too?
Yeah.
Yeah.
But she said we don't know yet, so maybe it's just an assumption.
My bulldog was a perpetual. If he wasn't sleeping, he was yawning.
And it would be interesting to see what yawning does to the locus ceruleus. Does that also calm and switch the locus ceruleus activity? Because it's interesting that facial nerve, like trigeminal nerve, through the vagus connects indirectly to the locus ceruleus and has a powerful effect on that.
The locus ceruleus chat. All right, nice. I got we're gonna be wrapping up here soon, guys. I appreciate everybody who has tuned in and listened up to this point. If you're still here, go ahead and drop a like on the video and a comment, a W in the chat. Let me know you're here and what your favorite part of the episode has been so far. But we're gonna get into two more articles that I found had interesting headlines. One of them's on nicotine and one of them's on smoking in general. And these are always, it's one of the many vices, people either have. It's going to be smoking or drinking, right? So we're going to dive a little bit into what smoking actually does to your immune system. How it actually impacts us. Because, we've always heard smoking is bad for us, but, and it's not good for your lungs. It's not good for your breathing, but let's go into it. Let's find exactly why this is the case. All right. So persistent impact of smoking on the immune system. Smoking not only affects immune responses in the short term, but also leaves a lasting imprint on the body's defense mechanisms through the milieu intérieur cohort of 100,000 or 1,000 healthy volunteers. The study identified smoking alongside latent cytomegalovirus infection and body mass index as a key factor influencing immune responses. This groundbreaking research demonstrates that the effects of smoking on adaptive immunity can persist for 10 to 15 years after cessation, attributed to epigenetic changes in DNA methylation that alter gene expression involved in immune cell metabolism. This insight opens new avenues for understanding how lifestyle choices, all right, like smoking, can have enduring effects on our health. Smoking significantly impacts both innate and adaptive immune responses, with some effects persisting for up to 15 years after quitting. The study, we always hear about, cancer people, they're like, oh, or not cancer, well, people who smoke cigarettes who got cancer, like, I quit smoking cigarettes. Well, you're not going to get it immediately. It's going to take a while for some of these effects to actually take place negatively in your immune system and for you to get an immune response, right? So the study used the milieu intérieur, I don't think I'm saying that right. It looks French though, intérieur cohort to demonstrate how smoking, latent cytomegalovirus infection, and body mass index are major factors affecting immunity. Long-term effects of smoking on immunity are linked to epigenetic changes, specifically DNA methylation, highlighting the durable influence of smoking on the body's defense systems. Like other factors such as age, sex, and genetics, smoking has a major impact on immune responses. This is the finding recently made by a team of scientists at the Institut Pasteur using the milieu intérieur cohort of 1,000 healthy volunteers established to understand variability in immune responses. In addition to its short-term impact on immunity, smoking also has long-term consequences. For many years after they have quit the habit, smokers are left with effects on some of their bodies.
Type 1 diabetes can happen at any age. Are you at risk? If just one person in your family has type 1, you're up to 15 times.
Shut up, ad. Gotta love it. All right, where we at? All right, so for many years after they quit, the smokers have been left some of their body's defense mechanisms acquiring while smoking. So how does this take place? How are these effects happening in our body? These findings, which for the first time reveals a long-term memory of the effects of smoking on immunity, which we published journal Nature on February 14, 2024. Individuals' immune systems vary significantly in terms of how effectively they respond to microbial attacks. But how can this variability be explained? What factors cause these differences? To answer this question, we set the milieu intérieur cohort comprising 1,000 healthy individuals aged 20 to 70 in 2011. Comments Darrow Duffy, head of the Translational Immunology Unit at the Institut Pasteur and last author of the study. While certain factors such as age, sex, and genetics are known to have significant impact on the immune system, the aim of this new study was to identify which other factors had the most influence. Scientists exposed blood samples taken from individuals in the milieu intérieur cohort to a wide variety of microbes, viruses, bacteria, and observed their immune response by measuring levels of secreted cytokines. Using the large quantities of data gathered for individuals in the cohort, the team then determined which of the 136 investigated variables, body mass index, smoking, number of hours of sleep, exercise, childhood illnesses, vaccinations, living environment, etc., had the most influence on the immune responses study. So there are variables stood out. 3 variables stood out: smoking, latent cytomegalovirus infection, and body mass index. The influence of these 3 factors on certain immune responses could be equal to that of age, sex, or genetics, points out Darrow Duffy. As regards smoking, an analysis of the study showed that the inflammatory response, which is immediately triggered by infection with a pathogen, was heightened in smokers. And moreover, the activity of certain cells involved and immune memory was impaired. In other words, this study shows that smoking disrupts not only innate immune mechanisms, but also some adaptive immune mechanisms. A comparison of immune responses in smokers and ex-smokers revealed that the inflammatory response returned to normal levels quickly after smoking cessation, while the impact on adaptive immunity persisted for 10 to 15 years, observes Daryl Duffy. This is the time it has been possible to demonstrate the long-term influence of smoking on immune response. Basically, the system appears to have something resembling a long-term memory of the effects of smoking. But how? When we realized that the profiles of smokers and ex-smokers were similar, we immediately suspected that epigenetic processes were at play. So I'm guessing they were talking about specifically cigarettes here in this study. No mention of marijuana, but I'm sure that they both have, their different type of effects on the system, on the immune system. So when we realized that the profiles of smokers and ex-smokers were similar, we immediately suspected that epigenetic processes were at play, says Violaine Saint-Andre, a bioinformationist, mathematician, bioinformatician at the Institute last year, the Translational Immunology Unit, and the first author of the study. We demonstrated that long-term effects of smoking on immune responses were linked to differences in DNA methylation, with the potential to modify the expression of genes involved in immune cell metabolism between smokers, ex-smokers, and non-smokers. It therefore appears that smoking can induce persistent changes to the immune system through epigenetic mechanisms. This is a major discovery elucidating the impact of smoking on healthy individuals' immunity and also, by comparison, on the immunity of individuals suffering from various diseases, concludes Violaine Saint-Andre. So, another interesting article about smoking in the immune system. Some research there kind of gives us a deeper look into the mechanisms that smoking are actually doing that are causing the immune system damage that smokers have. So we're going to get into a nicotine study, and we're going to close it off with a nicotine research video. We are currently at the 1 hour and 32 minute mark here. It's currently 3:47. I am over here in the West Coast. I am actually getting ready for 4 o'clock fantasy football draft here. So we're going to be wrapping up this podcast here soon, but I appreciate everybody who is. Stay tuned with me throughout this process, and obviously you're getting value out of this, and you see that some of this information is very interesting. At least I'm finding it interesting. So I hope that you are feeling what I am about learning, in a way about some of these topics like I am, because some of this stuff is interesting. So nicotine's influence on reward and aversion, posted February 13th this year, 2024. A new study delves into how nicotine influences the brain, revealing its complex effects on areas related to reward and aversion. Focusing on the medial habenula, researchers found its activity varies significantly with nicotine dosage, presence of menthol in the subject's sex, a discovery that challenges previous understanding of nicotine's uniform impact on reward pathways. Utilizing advanced techniques like patch, clamp, electrophysiology, and fast-scan cyclic voltammetry, the study offers new insights into nicotine's nuanced relationship with the brain, emphasizing the importance of considering individual differences in nicotine addiction research. Now, me personally, guys, I'm not much of a nicotine person. Like, I know a lot of people need their nicotine, and it's one of those things that they can't do without. personally, I don't really like the feeling of nicotine. Like, I'm kind of alert and present as it is. So too much nicotine just gives me a headache. I've tried one cigarette in my lifetime. I tried to puff on a cigarette when I was like 16, and I didn't. I was maybe 14, 15, 16, and I just never was my thing, honestly. But I'm gonna get into it. The study highlights how nicotine's effects on the brain are influenced by dosage, menthol presence, and sex, particularly in the medial habenula, a region associated with nicotine aversion. Unlike assumed uniform effects on reward centers like the ventral tegmental area, the MHB shows varied activity, underscoring nicotine's complex influence on brain function. Future research also aims to explore the aversive pathway of nicotine usage further, focusing on the interaction between the MHB and the interpundicular penducular nucleus to understand withdrawal symptoms and intake limitations. A new study led by researchers at the Marshall University Joanne C. Edwards School of Medicine sheds light on the intricate interplay of brain regions involved in nicotine's effects on the human brain. The research, published in eNeuro, a open-access, peer-reviewed scientific journal published by the Society for Neuroscience, explores how nicotine influences key areas associated with reward and aversion, showcasing a nuanced relationship that varies based on dosage, sex, and distinct brain regions. So it's going to affect everybody different. So nicotine obviously is not for everyone, and it affects everyone different, as I've realized. The medial habenula, a region known for regulating nicotine aversion, takes center stage in the study. Researchers discovered that MHB activity experiences fluctuations, either heightened or diminished, depending on factors such as the amount of nicotine consumed, dosage variations with or without menthol, and the sex of the subject. Intriguingly, this modulation was not mirrored in reward centers like the ventral tegmental area, challenging previous assumptions about nicotine's impact. The study demonstrates that the activity of crucial brain regions associated with nicotine dependence is altered in different ways based on nicotine dosage and sex, said lead researcher Nathan Olszewski, a biomedical researcher doctoral student at Marshall University in the laboratory of Brandon J. Henderson. Nicotine usage affects individuals uniquely, making it advisable for users to exercise caution. The study employed a vapor inhalation model of nicotine self-administration in mice, utilizing nose poking to earn nicotine vapor deliveries. So they poked these poor mice in the nose and injected it with nicotine vapor. Employing patch clamp electrophysiology, researchers elucidated changes in neural excitability in the medial habenula and ventral tegmental area based on nicotine dosage and sex. Fast-scan cyclic voltammetry was also used to assess changes in dopamine release dynamics in the nucleus accumbens. Okay, in our field, attention has predominantly focused on specific regions like the ventral tegmental area, said Henderson, an associate professor of biomedical sciences at Marshall University. This study underscores the necessity of exploring other brain areas controlling the negative aspects of nicotine exposure. The researchers aim to expand the investigation to other brain regions, particularly focusing on the interplay between the MHB and the interpundicular nucleus, or IPN. The circuit, known as the aversive pathway of nicotine usage, plays a crucial role in limiting nicotine intake and withdrawal symptoms. Future research will utilize electrophysiology, confocal microscopy, and RNA fluorescent in situ hybridization, or FISH, to understand how nicotine alters the activity and expression of nicotine acetylcholine receptors in this aversive circuit. Okay, so now we're seeing that nicotine does influence everyone's brain differently and in many different ways. So this last little video I got for us today to react to is going to be called it's a little YouTube video by Medical Dialogues called Study Shows Nicotine's Varied Effects on Brain's Reward and Aversion Center. So this is directly related to the study we just read there on nicotine. So let's see what this video has to say. Let's get right into it. It's a 3-minute video. We're gonna make it quick, and yeah, let's jump right into it.
Hello and welcome to Medical Dialogues. I'm Anshika Mishra. Today we'll talk about study shows nicotine varied effect on brain's reward and aversion centers. A new study led by researchers at the Marshall
Chat, can y'all understand her?
University Joanne C. Edwards School of Medicine
She's got a thick accent.
highlight the intricate interplay of brain regions involved in nicotine's effect on the human brain. The study published in the open access journal eNeuro by the Society of Neuroscience examines nicotine's complex effect on brain regions tied to reward and aversion. It highlights the nuanced interaction that is influenced by factors like dosage, the presence of additives such as methanol, and the sex of the individual. The focus is on medial habenula, that is the MHB, critical for nicotine aversion, where researchers observe variable activity levels based on these factors. This variability contrasts with the behavior in reward-related areas, such as the ventral tegmental area, offering new insights into nicotine's differential impact on brain. This study demonstrates that the activity of crucial brain regions associated with nicotine dependence is altered in different ways based on nicotine dosage and sex, said lead researcher Nathan Olszewski, a biomedical...
Yeah, guys, as we just read, nicotine affects especially male and females differently, and there's also different factors, which I didn't know. That's interesting.
Research doctoral student at the Marshall University in the laboratory of Brandon J. Henderson, PhD. Nicotine usage affects individuals uniquely, making it advisable for users to exercise caution. Study
goes to show women and men are not the same.
Employed a method where mice inhale nicotine vapor by poking their nose into a device, mimicking how humans might self-administer nicotine.
Are you also aspiring to excel in medical journalism and delve into the
Yeah, guys, it just seems like she's just pretty much talking about what we just read about. So yeah, guys, that's it for the reactions for today. Guys, it has been another amazing episode. We're gonna be wrapping up here soon. I got some fantasy football drafts coming up here. Wish me luck. I got 2 $5 leagues for a total of reward of $50. So 2 $5 buy-ins. I think I'm gonna do about 10 to 15 teams. So we got drafts here at 3 and 4. Trying to get all these drafts in before the season starts. I feel like that's the best way to get into a draft is before, so that it's kind of no cheating and there's no way of knowing what players are going to do good on the season, getting into it like in the middle of after the season already started. You kind of want to. It takes more skill to make proper guesses, picking your draft and everything like that. So yeah, everybody. I just want to thank everyone again for joining. If you haven't yet subbed up on YouTube, go ahead and hit that subscribe button. On Apple Podcasts, Spotify, go ahead and hit that like and give me a 5-star review. Thank you everyone again. I love you guys. We're going to be doing some more podcasts coming up here soon, at least once a week, sometimes even twice a week. And other days we'll be streaming on Rumble and Twitter. So make sure you're subbed up on there. We got some guests here coming up soon too. Some special guests, so stay tuned for that. I can't wait to reveal it to you guys. We got some, and we're going to be doing different topics and different type of episodes here coming up soon, different type of interviews. Episodes like today is kind of more so just pulling up some news, seeing what's going on in the world, and expanding our knowledge and just learning more and and I always find these type of podcasts interesting as well. But got a lot more different things coming up for y'all guys, so stay tuned. We got some last minute chatters in the chat. Shout out to Joe in the chat. He says, "What you think? What you think Trump 2024?" Hey, brother, we on YouTube. I really don't want to get into it too much. Just know, we rocking with Trump over here. I'm not vote. I can't see myself voting. In general, I don't think I'm gonna pull that trigger. I kind of not really into politics like that. Like of course, I'm gonna, go for who's ever better for the country, right? And right now, it's looking like Trump's got stronger points than his opponents. I'll just say that. Let's see. I'm rocking with Trump right now. I mean, I didn't vote last year. Not gonna lie, I just didn't. But this year, I see myself doing it. So yeah, let's wrap this up. GL with your draft. Appreciate it, brother. So yeah, guys. Before I log off here, guys, like I said, follow up, support on the IG, Instagram on at IcyMick, Facebook. We got the page, the Stormy and Friends page. And then all the other social medias will be at the bottom, especially the Patreon, guys. Go and sub up on the Patreon. We are getting content up on there every week, every day. So make sure to go check that out. And yeah, guys, I look forward to seeing you guys in the next one. It's been another episode, the StormCast here on a beautiful Sunday. I love y'all. Let's go. Bow.